Hi, my name is Olivier Pardo, and I lead the Cellular Regulatory Network Lab at the Division of Cancer at Imperial College. And today I'm going to tell you about two new therapeutic targets, ATR and CDK4/6, that we've identified for the treatment of methotrexate-resistant choriocarcinoma. So first of all, what are ATR and CDK4 and 6? ATR acts as a DNA damage sensor, and it then inhibits DNA replication and mitosis in order to promote DNA repair. On the other hand, CDK4 and 6 are cyclin-dependent kinases that are responsible for the progression through the G1 phase of the cell cycle, and it does that by phosphorylating members of the retinoblastoma family. However, these two targets are not completely separate, because in the cell, regulation of the cell cycle through CDK4/6 and DNA repair are interconnected. So the two processes need to work together in order to ensure cell survival. Our main question was, what do we do with methotrexate resistance in choriocarcinoma, and what new therapeutic target could we find? And in order to answer this question, we used two cells types, the JEC3 cells, which are methotrexate sensitive, and the JEC3R cells, which are the methotrexate resistant counterpart. And as you can see here, the sensitive cells respond much more profoundly to methotrexate than their resistant counterpart. And this, in part, is due to the overexpression of the target for the methotrexate, which is DHFR. However, I say partly, because if we downregulate DHFR in the resistant cell, we only partially recapitulate the sensitivity to methotrexate. And therefore, this suggests that there are other molecular targets that regulates the resistance to methotrexate in these cells. In order to identify these targets, we went on and did a proteomic profiling of the resistant cells versus the sensitive one. We looked at proteins that were differentially expressed and grouped them functionally. And what you can see is that one of the biggest functional group, module 0, was associated with proteins that regulate cell cycle and DNA damage response. We therefore got interested more into these two pathways. And here you can see in color coding, red meaning overexpression or increased phosphorylation, and blue downregulation. We looked at the expression of the proteins along the cell cycle. And what we can see here is that while the beginning of the cell cycle at the G1 phase seems to be promoted with downregulation of an inhibitor of cell cycle progression and increased expression of CDK4, the later part of the cell cycle seemed to be completely inhibited with increased expression of P21 and P27, two inhibitors of cell cycle progression, and decreased expression of cyclin-dependent kinase 1, which promotes the entry into mitosis. And this results into a delay in the cell cycle in the methotrexate-resistant cells, with an increased proportion of cells in the G1 phase of the cell cycle and a decreased proportion of cells doing DNA synthesis. This results in the methotrexate-resistant cells proliferating far slower than their sensitive counterpart. These changes that we observe in choriocarcinoma are not limited to choriocarcinoma, and in fact these type of changes are reproduced in other types of cancer where there is methotrexate resistance, as we've been able to demonstrate by again comparing sensitive and resistant cells from other tumor types. The thing that caught our eye here was the downregulation of P16 and the increased expression of CDK4. And this is because in other cancer types, such as breast cancer, these qualified patients for an inhibitor of CDK4, which is called palbociclib. And therefore, we tested this inhibitor in resistant choriocarcinoma setting. And what we can see here is that the resistant cell line is far more sensitive to palbociclib than their sensitive counterpart, while normal placental cells seem to be completely resistant. And this provides us with a very nice therapeutic window. Therefore, we tested this inhibitor in vivo, and administration of palbociclib was very efficient at decreasing tumor growth at early and late time points. However, palbociclib was unable to sensitize the cells to methotrexate. So their sensitivity to palbociclib was associated with a methotrexate resistant status, but was not linked to that status, which was quite interesting. We then looked at the changes in the DNA repair pathway, because that was part of the biggest module that we had identified. And in particular, we looked at the ATM and ATR pathway, which are the two main pathways responsible for DNA repair downstream of DNA damage by chemotherapeutic compounds. And as I hope you can see here through this color coding, the ATM pathway seems to be majorly switched off in the methotrexate resistant cells, while the ATR pathway appeared to be promoted. And this resulted in the increase in homologous recombination and non-homologous end joining, two main pathways of DNA repair in the resistant cells versus the sensitive cell, with maybe the non-homologous end joining being the primary increase in DNA repair in these cells. Because of the increase in the pathway for the ATR side of DNA repair, we wondered whether ATR was actually linked to this increase, and we answered this question by downregulating, by knocking out ATR in the resistant cell line. And this showed us that this canceled out completely the increase in non-homologous end joining that we saw in resistant cell lines. On the other hand, it didn't do any change in terms of the homologous recombination increase. And so it shows us that ATR is at least partially responsible for the increase in DNA damage repair in the resistant cells. So we then wondered whether this could sensitize the cells, taking out ATR could sensitize the cells to methotrexate, because methotrexate will lead to DNA damage. And here we used two separate knockout cell lines for ATR in the JEC3R setting. And you can see that both clones showed sensitization to methotrexate as compared to the wild type cells that do express ATR. But as I showed you in the diagram in the previous slide, ATR work as part of a pathway that contains two other kinases that are downstream to ATR, CHEK1 and WEE1. And what we've been able to demonstrate using small molecule inhibitors this time is that inhibiting either of these three kinases leads to the same effect, which is sensitization to methotrexate. Here you have inhibition of ATR, inhibition of CHEK1, and inhibition of WEE1, sensitizing the resistant cells to methotrexate. Because the ATR inhibitor is in clinical testing for other types of cancer at present, we decided to now test this inhibitor in our in vivo model for methotrexate resistance. And as you can see here, the use of ATR inhibitor alone significantly prolonged the median survival of the animals with methotrexate resistant tumors. However, disappointingly, in view of what we had found in vitro, we did not find that the combination of methotrexate and ATR was any superior in vivo as the use of the ATR inhibitor on its own. So at this point, it seems that just targeting ATR could be in itself a new therapeutic strategy for the treatment of resistant disease. So in conclusion, what I showed you today is that resistance to methotrexate in choriocarcinoma is associated with a delay in cell cycle progression and an increase in DNA damage repair. And this provided us with two new therapeutic strategies: inhibition of CDK4 and 6 with palbociclib that inhibits the growth of both methotrexate sensitive and resistant cell lines, but more prominently in the resistant setting, and inhibits the growth of methotrexate resistant tumor in vivo. However, it does not lead to sensitization to methotrexate, either in vitro or in vivo. On the other hand, inhibition of the ATR pathway sensitizes methotrexate resistant cells to methotrexate in vitro, but not in vivo, where, however, inhibition of ATR on its own is sufficient to delay tumor progression and increase survival in our in vivo model. So this work was spearheaded by Marina Georgiou, Penny Ntevelu, and William Stoke in my lab. And here you've got a recent picture of my lab members. But this work could not have been performed without our close collaboration with Kevin Elians, Neil Horowitz, and Ross Berkowitz at the Dana-Farber Institute. So I had only a limited amount of time to present to you our data. If you want to know in more details other things that we've done in our research, please refer to our recently published paper in Oncogene from this year. Thank you very much for your attention, and I'll be happy to take your question now.